El dice sí, aunque no tenga nada conectado, porque no tiene ningún micrófono conectado.
Okay, so welcome everybody to today's seminar. We have the pleasure today to have Laia Pérez Sorribes. She is doing the PhD supervised by José Antonio Godoy, and she is going to tell us about her and then about the genomics of the Iberian lynx. So welcome Laia, and the floor is yours.
Thank you, Isa. As Isa said, I'm doing my PhD with José Antonio Godoy. I just started my second year. And my PhD is called Genomic Basis of the Genetic Rescue of the Iberian lynx. But first, I would like to introduce myself. I'm from Barcelona, where I studied at the Autonomous University of Barcelona, my degree in environmental biology and my master's in terrestrial ecology and biodiversity management. I did my practicum and my master thesis with Salvador Carranza in the Institute of Biology Evolutiva. And I also, at the same time, co-founded and collaborated in a local NGO working for amphibian and reptiles conservation, environmental education, and doing some local research. Thanks to all of that, I got a scholarship from the Societat Catalana d'Herpetologia to do a fieldwork project on salamanders. And then when I finished my master's, the COVID came. I started working at the Universitat of Barcelona on population genetics and molecular ecology, doing a lab technician, but also helping with a lot of stuff. And then, as it was part-time job, I combined it with science communication for the Catalan Biogenome Project. And I got a short-term scientific mission grant to go to KU Leuven with Professor Joachim Merguet to do a short stay there in Belgium and in genomics. And when I came back into Barcelona, I've been in the Institut Botanic working with Gerard Talavera, also as lab technician. When I received the notice that I got the PhD grant to come here to start my PhD on lynxes. And I also enrolled the GNOA, that it's a continuation of GBIKE, cost action. So now, focusing on my PhD, I would like to start speaking about small populations. Small populations have are in danger because of can suffer from environmental stochasticity, but also there are genetic mechanisms that are acting to this population. So when you have small populations, genetic drifts can act and reduce the increase the inbreeding, reduce the diversity, and then reduce the adaptive potential of the population. And if it reduces fitness, we are facing inbreeding depression. So this is getting worse and worse every time. So this is a booklet. So how is the way to escape from this? Is introduce a new gene pool. It can be due to gene flaw or to translocations of individuals that reduces the inbreeding increases the diversity, and this is called, when it happens, genetic reinforcement. And then if it's the adaptive potential increases and the fitness increases of the individuals and the population grow, then we are facing genetic rescue. So then I would like to start by explaining some concepts that will be important for understanding the chapters of my thesis. That it's ancestry here, or called theta S as well. We have two parental lineages that could be two populations. Then when these populations just admix, just mate, the result is an F1. And how is this genetic material translated to the descendants? You have 0.5 of each of these ancestries, from the father and the mother. And when you have F2, you as well have 0.5 of this parental lineage and this parental lineage. When we talk about interclass heterozygosity, here we are not talking about ancestry, we are talking about heterozygosity. So we have the two parental lineages that have their own ancestry. And then when you have F1, you get one copy of the father and one copy from the mother. So you have that all the genome is heterozygote on this. So then when you have an F2, you also have, let me explain. Yeah, here you have also 0.5 as the other one, but here we focus on this, that is heterozygote, this part, but this is homozygote. So now, why is this important? Because when you mix two ancestries, you can have deleterious mutations that when you have the F1, these deleterious mutations, the recessive deleterious mutation, is not expressed, so this F1 would be fitter than the parental lineages. And that is called a hybrid vigor. So, because you can have these two parental lineages, here you have the mutations that are homozygote, but here when you have the F1, these mutations are not in the same position, so these mutations are not expressed, so this one would be fitter. So now it's the time to talk about the protagonist of my thesis, that is the Iberian lynx, who is an endangered species. Well, it was endemism of the Iberian Peninsula that faced an historical decline. It was one of the felids most endangered in the world in 2002. We only had two populations with less than 100 individuals. So then the Life Project started to act. Here you have the different projects that have been applied. It has been done in situ management, captive breeding, and reintroductions, genetic reinforcements, and now we are doing a genetic monitoring of these populations. So we are now is vulnerable, is still in danger, but we are better. So these captive breeding and reintroductions have been applied the criterion of maximizing the diversity and reduce the consanguinity individuals. So yeah, we had the two populations that we were talking before. So Doñana and Andújar. This is the original, well, the ancestral gene pool of the Iberian lynx. We've seen here that Doñana population diverged here and has not had gene flow between the other populations. These other lines here are the populations that went extinct. And here we have the other one, Andújar, that is more similar than the original gene pool. We have that these two ancestries, we will call the Doñana and Andújar, have accumulated different genetic diversity. So there are alleles that have been fixed in both populations, and also not only fixed, but also the frequencies, the allele frequencies are different between them. So here I just done a small test of FT1 SNPs that they are differ between the two populations. Here you have the points of these SNPs. They are around the whole genome. These are the chromosomes of the lynx. And we have like around 15,000 SNPs. So here's just to remember a bit of the schema of before. So in the Iberian lynx, we have evidences of inbreeding depression. We have a lot of incidences on genetic disorders. Here you have some of them. And we also have evidences of anormal sperma. But everything is not bad. So until the population was decreasing from past, but in 2007, this is the Doñana population. Here you have the diversity, genetic diversity was decreasing, but in 2007, some individuals from Andújar went translocated to Doñana. So this genetic diversity increased, so we have genetic reinforcement, and it's also translated into the demography. The demography also increased. So then we are facing genetic rescue. We are going to test with numbers to demonstrate that this is happening. So the global objective of my thesis is to study the genomic basis of this genomic rescue in the Iberian lynx. So for studying that, we need two types of data. We need genomic data and we need fitness data. So I will start by talking about which genomic data we are generating. We are extracting DNA from blood samples and from muscle samples. We are constructing ourselves the libraries in the lab, and we sequence the libraries in Novogene, and then we do the data filtering and imputation. Imputation, what's that it's performed? Well, we constructed two types of libraries. We construct the high coverage library here for 50 individuals, and then that will be our reference panel, and then we will have this for 300 or 400 individuals. We will see at the end how it goes. Now we are constructing this, so we are sequencing in low coverage, and then fill the gaps that are not sequenced with like a puzzle with the high coverage panel. And we will check for Mendelian incompatibilities with the pedigree of the individuals. So thanks to a lot of work from a lot of people for a lot of years also, we have fieldwork data available of yearly survival, reproduction, and also thanks to genetic monitoring that it's being performed here. Here's how it works. We also had census data and genealogy that we can obtain the reproductive, the exit of the reproductive of the different individuals. So now the chapters, my thesis is divided in 4 chapters. The first block is on the genetic rescue that is facing Iberian lynx right now. First is to evaluate the effects of these translocations on fitness recovery. Then look into the different regions on the genome to look about local ancestry patterns and deviations of neutrality. And the second block is to assess how how other natural processes can help in restoring genetic diversity. So first, the first chapter would be this, to evaluate the effects of translocations into the fitness recovery. Here we will use genetic variables and fitness variables to do some generalized linear mixed models to evaluate this, what are the effects of genetic variables in fitness of these populations. We have some preliminary data obtained by microsatellite data, and with few generations, now we will have more generations, more data, fitness data, and more genomic data. So we've seen that F1s seem to be fitter than the parental lineages. So then in the second chapter, we will look at the genetic regions associated with this fitness increase, looking for deviations. We want to look if there's an increase of ancestry in each of these regions. So we are going to infer the local ancestry patterns, do detection deviations from neutrality with genomic lines and gen drop analysis. So here we would like to test about hybrid vigor for all of these genetic positions, regions. So here is how it would look a bit. We have the ancestry that I was talking in the beginning, and interclass heterozygosity. You will expect the pure individuals here and here, and then the F1s, you will expect 100% of interclass heterozygosity and 50% of hybrid index, that ancestry, and the F2, and the other ones should be around here, the back crosses and the other ones. If they are here, are deviated from the neutrality. So we will trying to understand what's happening there. So then going to the the third chapter, this is not Iberian lynx, this is boreal lynx. These species we know that have been overlapping the habitats. So, and we have evidences of introgression, of past introgression. We know from previous studies of the group that gene flow has left some genetic material in the Iberian lynx genome. So we know in which regions are this happening and in which direction it went. So now we would like to assess if this was beneficial for the Iberian lynx, this introgression. So we would like to assess the signals of adaptive introgression in the Iberian lynx genome. We are going to do it with Volcanofinder, that it's an analysis that scans your genome and try to find this kind of patterns that is like a volcano. If you see, like the crater here, this is the signal for introgress regions that the diversity increases, and then when there's selection, the diversity decreases. So we are going to look for this kind of patterns in the genome. And the last chapter, we are talking with Dr. Aida Andres in London. It's under construction. She is expert in balancing selection, and we would like to assess how this balancing selection can contribute in sustaining genetic diversity despite drift. So we are going to take the population previous to the bottleneck and past the bottleneck. We will obtain the data from the Andújar as non-bottleneck and Doñana as bottleneck. We would like to find which regions are under this balancing selection and compare it between them. So I just forgot to explain about what's balancing selection. Balancing selection is a type of selection that benefit the polymorphisms. So you will try to find high genetic diversity in certain regions, ancient polymorphisms, and excess of heterozygous. So with all of that, I would like to thank my team, all the people that has been working in the previous projects before, that thanks to them, I could be here, and thank you.
Thank you, Laia, for this wonderful talk. It's time for questions. I'm sure that there should be some questions. If there are not, I have. So any question?
Well, thank you very much for the presentation. It was very interesting. And, well, you have a lot of work ahead of you. But there was one aspect that worries me. And I don't know, maybe I misunderstood. But when you are talking about imputation, you are going to work with low coverage sequence. That means that for some other regions of the genome, you are going to infer what that should be. And inferring what that should be means that you should not trust any mutation that you identify, any possible non-synonymous change, any deleterious mutations. You need to be able to track what areas of the genome have been imputed, or maybe imputation is not for that. Isn't that going to be a problem? Wouldn't it be better just to try to work with the raw sequences without trying to reconstruct the whole genomes and maybe doing more subsets of the genome, or working at sample per sample? I know it's a lot of work, but I don't know if it's dangerous to impute, to infer what the mutation should be.
Yeah, we have the reference panel is being selected to have the most diversity of the lynxes. So we have individuals from captivity, and pure individuals from Andújar, from the reintroduced populations, and was specially designed to maximize the diversity. So in theory, we will have a better approach in putting this. Lucia as well, she is doing tests to know how the imputation works, if it works good or not. And for now, she's obtaining good results. So she could explain maybe better than me. Yeah, it's true that if you have, it's always better to have higher coverage, for sure. But in this way, we try to sequence more individuals and less cost, because it's a trade-off, cost and data. So that's why we are working right now.
But I guess that you will have to be very careful with the deleterious variants that you identify, which ones are real and which ones are not.
Yeah, we also can track which regions are inputted and which not. So if we detect something, we will take a look on it.
Thank you.
You're welcome.
More questions?
Hi, Laia.
Hi.
I enjoyed your talk. I have a couple of questions. After translocation, how many generations should we wait to can evaluate the potential effects of a genetic rescue?
Well, if we're the hybrid vigor, just the F1, but it's always better to have more generations to see what's happening. We have right now, I think it's 6 generations, if I'm not wrong. So it would be better to have more, but it is what it is. We cannot reproduce lynxes more. So we will see what we get. I don't know which point we'll try to do what is in our hands. I don't know.
Yeah, because it could be easy for flight, for example, but could be challenging for large mammals. I understand. And which could be the negative effects of the translocking individuals of different populations? But I think could be some.
Yeah, there are, but few examples that this happened. It's called outbreeding depression. When this happens, that the hybrids are not better, they are bad, they are not successful, but in this case it's going good. You have to, there's a schema, I remember, I don't know in which paper, but it tells you, if you have this kind of individuals and you have like diverged less than, I don't remember the numbers, but 100 years, and if you have this genetic diversity, you should do this, you should do the other way around. You can, if you want, I can check it because I don't remember which paper it was, but I can send you later.
Thank you. I have a last question. I'm wondering which should be the size of the population to start a similar program, to start to a genomic rescue program. So at what population size we should start?
I don't know in this case what critical that we were losing the species, but I'm not expert on this right now.
I think it could be different across species, but I wonder if there is a potential.
Well, they say that the NE is there 500, but I don't know in which point you should do that. You also have to evaluate the other thing, the schema that I was talking before, to avoid outbreeding depression. That this happens when you have two populations that have been evolving separately, and they have been accumulating some adaptative regions and things. So that's all I can tell you.
More questions? Yeah.
Hi. Very nice talk, Laia. Really nice project. I was wondering, you said that you had microsatellite data, which has done before with fitness. Are some of those individuals that you have microsatellite data being analyzed with the whole genome approach as well? Or
I haven't analyzed anything yet. Okay. So for for my side, no. But Laura is mostly sequencing all of the individuals of the Iberian lynx with the genetic monitoring SNPs. So I think we can just re-evaluate it with nice. More data. Yeah.
And then for chapter 3, you said that, well, once you look at the areas that might have the potential adaptive introgression, are you like doing trying to identify which genes are involved in this area, and what is your approach for doing that? What are you planning to do?
Yeah, to do go there. What are the regions involved in? Yeah.
More questions?
Hi, thank you. So I think that demographic stochasticity, so the random death of individuals, is still a thing in lynx populations, right? Because
of
numbers are
road kills and things.
So in a thesis like that on genetic rescue, so I wonder, how can you differentiate that the genetic enrichment of populations are contributing to the population dynamics, or other things are taking place here? Because it seems that earlier you said that thanks to the increase in genetic diversity, populations are increasing. I can see that straight line as you see it. I think that there are many moving parts here.
Yeah.
How can you really be sure that the genetic efforts you are doing are really impacting the populations? Because extinction is a demographic thing, and expansion as well. So actually the number of examples we have that genetics is the major cause of extinction, I'm not sure whether those examples exist. So it's a factor promoting that. So what's your opinion about that? That populations are still low, demographic stochasticity might be a major force driving these populations. How can you assess the value of genetic rescue for the lynx populations?
Well, now we are not sure that genetic is the only way, the only thing that is influencing this recovery. That's why my thesis. We are trying to infer it. But as well, you have, as you said, ecological things that are impairing. And the previous data that I showed with the microsatellite data that increases the offspring number, it was after the genetic rescue. There have been a lot of actions in the field, like feeding, supplementation, and anything was working. When the translocation came, then the population started to grow. So this is an indicative that maybe it's this. But we also would like to apply, if data is available, rabbit densities and this type of data that you are talking about into the GLMS tests to see.
More questions?
Thanks for the nice talk. That was really interesting. I just have a follow-up question, actually. I was wondering if you could look at some traits that are known to be affected by inbreeding depression, like sperm motility or something, in your F1, or, to see if the genetic rescue is having an effect on those traits, rather than just on the population, basically.
I think it could be interesting. It's not planned right now, but maybe, I don't know. But we have from the inbreeding depression with, here we have some graph about the normal sperma that was from previous analysis, but we haven't checked right now what's happening.
Because that was before the rescue or after?
Yes, this was before.
Okay, so my question is the last question.
Okay.
This was related with the number of generations. So,
yeah.
So I understood that you just have data from F1s and F2, right?
No, we will have more.
You will have more.
Yeah.
Okay, because it will be interesting to see if this genetic rescue is just in the very first generations that compared to all older, like more generations, because I think that there is a paper that at the very beginning after the genetic rescue is like the population goes up and suddenly then, it will be super interesting. I'd love to have that kind of data for wolves, but I don't have it. So thank you, Laia, for this wonderful talk, and thank you everybody for coming.